There are 5 primary targets of antibacterial drugs. Before we get to each of those in detail, let's take a look at some of the things the cell needs to do in order to carry out its daily existence. So we know that bacterial cells all possess DNA, and that DNA is used to control protein synthesis. We've discussed how the DNA is used during transcription to make mRNA, and during translation, that mRNA copy is used to make protein. Those cellular proteins can be enzymes, or they can be structural proteins, other enzymes or other proteins that we're not going to discuss here. So those proteins can be enzymes, and we've learned that enzymes control all of the metabolic pathways in the bacterial cell. Metabolism can be broken down into catabolism, which is ATP synthesis, or anabolism, which is the making of any larger molecules from smaller molecules, and this process uses ATP. Now, those anabolic pathways, there are many of them, but we are going to focus on a few: making cell wall, making cell membrane, making nucleic acid such as DNA and RNA, and also making protein is an anabolic process. The cell makes new DNA for replication or binary fission. And as long as the cell is allowed to do this, to use its DNA to undergo protein synthesis, carry out metabolic pathways, the cell is going to continue to exist happily. It's going to reproduce. It's going to make more bacterial cells. Some of those bacterial cells are bad. Those are the ones that we are concerned with. So we don't always want bacterial cells to be able to undergo life in this organized manner. If they're a bacterium that's making us sick, we obviously want to disrupt this process. So there are 5 primary targets of antibacterial drugs. The first type of drug is going to interfere with nucleic acid synthesis. Now, the drugs that interfere with nucleic acid synthesis might stop RNA synthesis, but they might also stop new DNA from being made as well. If the drug stops RNA synthesis, that means everything downstream of RNA synthesis is stopped as well. So we would not get protein synthesis, we wouldn't have metabolism occurring, the cell would not be able to make energy, it wouldn't be able to reproduce. Ultimately, this is going to kill the cell, and so these types of drugs are given the suffix "cytal." They're bactericidal because they are going to kill that cell eventually. And an example of a drug that interferes with RNA synthesis is something called rifampin. Now, drugs that interfere with DNA synthesis often are not cytal. They are what we call static, bacteriostatic, because they stop bacterial reproduction. So they don't necessarily kill the cell, but they do interfere with bacterial reproduction, because if the cell can't make new DNA, it can't undergo binary fission. And so an example of a drug like that would be a fluoroquinolone. The next target is to interfere with protein synthesis. And drugs that interfere with protein synthesis, most of them work by binding to the ribosome, and there's a great variety in how they can do this. Some of these drugs bind to the small ribosomal subunit, some of them bind to the large ribosomal subunit, some bind reversibly, some bind irreversibly. For the purposes of our class, we're just going to say most of these drugs work by binding to the ribosome. And then if you want to take a microbial physiology or a pharmacology class, you can get into those details. So these guys bind to the ribosome, and that prevents things like mRNA and tRNA from binding. So the mechanics of protein synthesis can't occur. Now this is going to bring us to another topic, and that topic is selective toxicity. One of the things we want our drugs to do is harm the bacterium, but not us. Okay, but we both have ribosomes. So how do drugs that interfere with protein synthesis harm the bacterium, but not us? Eukaryotic and prokaryotic ribosomes have different structures. So basically, if they have a different structure, the drug will bind to the prokaryotic ribosome, but not the eukaryotic ribosome. It does get a little dicey when we talk about mitochondria. There's a section in your book that talks about the endosymbiotic theory. Mitochondria are said to have evolved from bacteria that were ingested by a eukaryotic cell eons ago. Mitochondria have their own ribosomes, and those ribosomes share the prokaryotic structure. So for the most part, drugs that interfere with protein synthesis are selectively toxic. However, some do become toxic to the human over time as they are in the body in high enough concentration for a long enough period of time to get into the mitochondria and begin to affect those mitochondrial ribosomes. Will suffer the adverse effects of that. Okay, our next target is to interfere with metabolic pathways. And this is sort of a catch-all for all of the other metabolic pathways in the cell that aren't directly related to reproduction or energy synthesis. So anything that happens outside of reproduction or energy synthesis is dumped into this category. So we have drugs that interfere with other metabolic pathways in the cell, such as folic acid synthesis. And drugs that interfere with folic acid synthesis include sulfa drugs or sulfonamides. And these drugs maintain their selective toxicity because humans get all of their folic acid from their diet, not by making it themselves. Bacteria oftentimes will synthesize folate. Humans have to ingest it. That's why you have to be careful when you read how the mechanism of action works. It says it interferes with folic acid synthesis. That means it prevents folate from being made. It doesn't destroy folate that's already been made. So be aware of those nuances and be careful when you're reading how these things work. Take them at face value. Don't try to interpret beyond what it says. Our next target, I should have left a little more space here, is to disrupt cell membrane integrity. So this is not a good target because if you disrupt the cell membrane, we learned early on that bacterial cell membranes are similar to our cell membranes, and so these drugs that do this are going to affect us as well. So selective toxicity is very difficult to maintain here. We do have a few examples of drugs that work this way. One you may have be familiar with, polymyxin and the polymyxin B. These are found in topical ointments. We can use these because the upper layer of skin cells are dead and keratinized, so it shouldn't have much effect on our own cells if we use them for a short time as a topical ointment. That leads us to the first class of antibiotics discovered, and that is the drugs that interfere with cell wall synthesis. So group number 5, I saved the best for last. Now, again, pay attention exactly to the wording. Interferes with cell wall synthesis. These drugs only work when new cell wall is being made. They don't destroy existing cell wall. That means that these drugs only work during binary fission. That's why knowing the generation time of the bacterium that you're targeting is important. If it has a long generation time, if it doesn't go through binary fission very often, these drugs aren't going to work very well. If it's a bacterium that replicates very quickly, it has a short generation time, then these drugs are probably going to work fairly well for that bacterium. So there are different ways to interfere with cell wall synthesis. And the first class of drugs discovered are the so-called beta-lactam drugs. And these are penicillins and cephalosporins. And down here we can see an example of penicillin, an example of cephalosporin, and these drugs are called beta-lactam drugs because this structure right here is called the beta-lactam ring. That is the active site of these drugs. So before we can talk about how beta-lactam drugs work, let's back up a little bit and review the structure of peptidoglycan. I told you guys peptidoglycan would come back again and again and again throughout the semester. So if we go back and we remember that peptidoglycan is made up of alternating subunits of NAG and NAM strung together like beads on a string, and then the really cool, the really important part that makes it really strong is that these NAM subunits are cross-linked to each other. Now, how do you think that cross-link is made? Okay, it's made by an enzyme. So these cross bridges or glycan bridges are put in place by an enzyme. And that enzyme's name is PBP, short for penicillin binding protein. So during binary fission, the cell is elongating, it's making new cell wall. PBP is building these cross bridges. Well, what if this is a bad bacterium and it's giving you an infection and you don't want to be sick? So you go and the doctor prescribes for you a beta-lactam drug. Those beta-lactam drugs will bind to PBP instead of the cell wall or glycan bridge substrate. So the drug binds to PBP instead of the, let's call it bridge material, right? So then the enzyme builds bridges that are not functional. They fall apart. It'd be kind of like substituting real bricks in a building for styrofoam bricks. They would collapse. They would be crushed. And so when the cell wall or the peptidoglycan is made, we don't get the glycan bridges, and the peptidoglycan ends up being weak. I call it holy. It's got holes in it. And so then the cell lyses due to osmotic pressure. So the cell wall is weak, it can't maintain its structure, and the cell is going to rupture and die. There are some non-beta-lactam cell wall inhibitors. And those are things like vancomycin and bacitracin. Okay, so these are the 5 basic mechanisms of action of antibacterial drugs. I now want to switch gears a little bit and talk about drug resistance, because that is such a huge topic. Resistance to antibacterial drugs can emerge through spontaneous mutation. So it simply occurs in the population, or it can occur through gene transfer, that is, most often by conjugation and plasmid transfer. Now, the mechanisms of drug resistance include enzymes that degrade the drug. And a really good example of this is something called penicillinase or beta-lactamase. Penicillinase or beta-lactamase, actually, if we look at this picture, it destroys the beta-lactam ring. It's an enzyme that snips that ring structure, and then the beta-lactam drug can no longer bind to the PBP and interfere with cell wall synthesis. Sometimes the bacteria will change the shape of the target of the drug. For example, they could slightly change the shape of the ribosome so that the ribosome still functions, but the ribosome is no longer able to be bound by the drug to interfere with protein synthesis. They can develop something called efflux pumps, which actually bind to the drug and pump it out of the cell. they can actually alter their metabolic pathways. So if we give them sulfa drugs to stop them from making folic acid, maybe they'll switch and just start getting folate from their diet, from the environment, just like we do. So sometimes they can switch their metabolic pathways. They can more tightly regulate what goes into the cell and simply not allow the drug in. And some bacteria form biofilms, which make it so that the drug can't get to the cell. So these mechanisms of drug resistance can occur spontaneously through mutation, which is a sort of Darwinian evolution, survival of the fittest, or they can occur through gene transfer, whereby a drug that has genes for one of these mechanisms can pass those genes to another cell. And that's a big deal with this beta-lactamase, is that the beta-lactamase gene is on a plasmid, and bacterial cells do pass that particular plasmid around from cell to cell. They can pass the genes for the efflux pumps as well. And so they are sharing these genes, and because there are so many more of them and they reproduce so much more quickly than we do, that the resistance is emerging much faster than we can keep up with the drug development. That's why it's important to engage in activities that slow the emergence of antibiotic microbes. And you can read about those in your textbook, and we'll be discussing those more in class as we go along.